Hi everyone. In this video, we will discuss more about inheritance patterns and whatever else has been kind of understood since Mendel's ideas on inheritance. So, like many different rules, there are some exceptions to them. And some of these ideas are basically referred to as the extensions of Mendelian inheritance. So some of these topics we'll discuss in this video are in the realms of incomplete dominance, codominance, pleiotropism, gene environment interactions, epistasis, polygenic traits, and certain X-linked traits as well. Now, with incomplete dominance and codominance, what we mean by this is that sometimes in cases, um, the alleles of a gene are not entirely recessive or dominant, or not dominant over each other. And in incomplete dominance, what that means is that they end up with a intermediate phenotype where it's like these flowers where they are red and white, but then when crossed together, they end up with a pink flower for like the heterozygotes. So in this case, the ratio, instead of our regular 3-to-1 ratio, what happens is that you have a 1 to 2 to 1 ratio in phenotypes as well, because the heterozygotes have their own color instead of displaying the dominant phenotype. But then with codominance, what happens is that the heterozygotes end up with both phenotypes at the same time. So it's not dominant or recessive to the other. And an example for that one is the blood types, where A and B are dominant over O, but when we put them together like AB blood type, A and B are not dominant over the other. They are co-dominant, that they display both antigens. Now, an example of co-dominance is like these sweet little cows where they have this coat here where a brown one and a white one come together. They have a cow with this fur color, and what happens is that's the Rowan cow fur color. So what happens is that both the colors are visible in that cow. In incomplete dominance, what happens is it's like an intermediate. So like this black chicken and white chicken crossing together, they have blue chickens, but then when we have the blue chickens cross with each other, it happens that we have one black, one white, and two blue ones. So the blue ones are like an intermediate between the two parent chickens. Now, there are also certain genes that are pleiotropic, which means that that gene can influence many different traits. An example would be like the fibrillin gene, where any mutation within this mutation can lead to a syndrome called Marfan syndrome, where it ends up, um, since fibrillin is like present within different body systems, it can end up with like increased height and limb length, or like heart and circulatory problems, vision problems, joint problems, anywhere that can just like where the fibrillin protein is present, it can like cause issues in those genes themselves. Another example of pleiotropic uh, disease, like genes influencing traits, is cystic fibrosis. It's basically a gene that is a disease that is caused by a mutation in this gene here, the CFTR. And what happens is with this gene, it kind of plays in role in like a the mucus and sweat secreting areas, like our skin itself will become salty and sweaty. There's like thickened mucus in like the respiratory, digestive, and reproductive organs. And then there's like um, greater risk for infections and respiratory diseases. And then it's unable to digest and absorb certain nutrients, regardless of normal appetites. And then there could also be infertility issues in males. So now one other thing that can also affect the phenotype is what is in your physical environment. And one example of this is like phenylketonuria, which is basically an inherited disease that is caused by an absence of an enzyme that breaks down a specific amino acid called phenylalanine, and it's basically contained in most protein containing foods. Now, if these phenylalanine levels accumulate to like toxic levels, it ends up with like irreversible mental retardation and developmental issues within like the individual that has this issue. And what happens is that if the affected individuals are placed on a low phenylalanine uh, diet, they can actually develop normally. So typically the newborn babies are screened for PKU in and other diseases within like 1 to 2 days of birth to kind of uh, make sure that they are set for whatever they need to do and be on their specific diets. So now with another way that these genes can be affected is through epistasis. It's when one gene affects the expression of another gene product. So in this case, it would be like um, the H protein that's required for A and B blood type to kind of have their own little glycoprotein to kind of keep the antigens there. If the H gene doesn't exist, they can't actually produce that H protein. And what happens is that the normal A and B glycoproteins can't be placed within that cell surface. And what happens is that it cannot attach the actual components for the blood type itself and just shows up like it's like not, it's genetically A or B, but it ends up appearing as type O. And this is kind of referred to as like the Bombay phenotype. And now we have another set of traits that are called quantitative traits. These are traits that can basically occur on a continuum. They are not discrete traits. These are like weights or heights or skin color. Basically, they are encoded by multiple genes or alleles and basically referred to as polygenic traits. These genes basically have additive effects on trait expression itself, and the environment itself can influence what's happening with those traits themselves. So what do we mean by polygenic traits? Polygenic traits are specifically for traits that are involved in like contributing a small portion of what is happening with a trait itself. The more dominant alleles you have, the more intensely you would express that specific trait. So in this example, it would be like skin color or height. if the person with this genotype will have darker skin or be taller than someone with this kind of genotype. So more dominant alleles you have, the more expressed it would be. The population usually exhibits a bell-shaped kind of trait for usually like a normal distribution, depending on the region you're in. So why do we have these multi-gene traits? It's mostly because it kind of allows you to produce a diverse population that can adapt to any changes in your environment. So like a skin color, it's like it helps with certain areas where it can be affected by like the environment itself. So when we look at it, it's like with dark skin, we have it more prevalent in where the areas that are like closer to the equator where UV damage can like hurt you more in like like more sunlight, but still gets through for enough vitamin production. But then with the light skin side of it, it's like uh, is more prevalent when it's like a away from the equator more towards the poles, because the low levels of melanin that's being produced is because like it allows more sunlight to come through for vitamin production, that it's like if it's not enough sunlight, what happens is it can get in and give more like vitamin D and folic acid and uh, keep things properly running in your system. Now, what actually determines the idea that an allele is either dominant or recessive? Usually it basically is the fact that there are like a gain of function where basically the presence of the protein is more prominent in the population, but then only one dominant allele is needed to express that trait in some cases. Conversely, if we look at like recessive alleles, they are like a lack of function. So if you're missing a protein or it's not like fully functioning, it's not like a um entirely the case of like actually gaining the function more or lack of function. It's more like what's more prominent basically. So with like these individuals with brown eyes, or like brown eyes with like blue eye recessiveness, what happens is they have the brown eyes because the melanin pigment itself occurs in the iris itself. But then with with these recessive genes, what happens is that the blue eyes have no melanin in that pigment expression occurs, ending up reflecting the blue wavelengths rather than the brown ones. Now, thinking about all of this, from what we know from Mendel's rules and into like human pedigrees, what we kind of use this chart called a pedigree chart. It basically is used to analyze human process that basically occur and already exist. It kind of allows you to determine the genetic relationships between individuals, and it predicts the mode of inheritance of different traits. And what we see here is that so the squares are male, the circles are females. And then if they're crossed out like this, they are deceased. If they are uh, covered in like this, like colored in like this, or the uh, circles being colored in, that means they are affected with whatever disease it is. In this chart here, we are looking at the pedigree of a Venezuelan family with Huntington's disease. So there are many different types of different traits when we look at different pedigrees. one of the sets is called autosomal dominant traits, where they are basically affected in individuals that have at least one dominant allele. And if the individuals are homozygous or heterozygous for the trait, they will basically show the dominant phenotype. And the homozygous recessive ones do not show that trait itself. So, there are a couple of examples. One of them is like Marfan syndrome, what we were talking about earlier. That is actually an autosomal dominant disorder, with individuals with one defective fibrillin gene can basically have the full effects of the disorder itself. Another prominent example of autosomal dominant traits is the Huntington's disease. Basically, if you have a person that is affected as your family, you basically have 50% chance of inheriting that faulty gene and getting Huntington's itself. So what exactly is Huntington's disease? It is an autosomal dominant dementia related illness, basically characterized by rapid jerky movements, usually with the extremities. And the symptoms generally develop between the ages of 30 and 50, when the children or individuals may have already had children themselves. So, like in autosomal dominant traits, they are usually equally seen in like males and females. It goes through every generation that any affected offspring would have at least one affected offspring parent, and then any affected offspring that are heterozygous, if only one parent is affected, then it goes from there. Then there are unaffected offspring that are homozygous recessive. If one parent is heterozygous, about half the offspring will be affected, half of them will not be. And the autosomal dominant trait itself does not skip generations. So Huntington's disease basically starts off with the Huntington gene, which contains an extra set of sequences of trinucleotide repeats that basically results in a toxic protein that is expressed mainly in neural tissues. And that is just if there's like a polyQ region expanded, the Q here stands for the amino acid glutamine. And if there's like more glutamines in that region of the gene, what happens is the proteins end up accumulating too much to a point where it's no good. And basically, the mutated Huntington proteins end up forming aggregates or like clumps that ends up causing cell death when it gets too much to handle in the uh, cells and the brain itself. So now we come to what is referred to as like lethal alleles, where some gene alleles are lethal if an individual ends up inheriting both copies of the mutation. This mutation can basically result in abnormal proteins being produced at toxic effects during development, or basically have the complete absence of a critical protein in itself. An example of autosomal dominant examples where it's like Mexican hairless dogs, where they have one copy of the mutated dominant allele, which prevents hair follicles from forming, like this poor doggy over here. And what happens is that the normal version of H, basically, with two of them, have hair. So that genotype would be like like recessive, but the homozygous dominant ends up like lethal. So homozygous version of the both, it ends up with lethal alleles. So what to kind of keep them from like preventing lethality in these cases, they cross these dogs with hairy dogs, the poor hairless dogs, to get basically like a 50-50% chance of getting a set of dogs that actually can survive. So in that second generation, it would basically just be crossing them with hairy dogs and get half Mexican hairless and hairy dogs. So now we have what is called autosomal recessive traits, and this is basically when two recessive alleles and they are homozygous recessive. So basically, the individual's parents are likely to be heterozygous carriers for that specific trait, but do not actually show the phenotype itself. And individuals with like two different alleles or dominant alleles and do not have or pass on that recessive trait. So, for example, cystic fibrosis is autosomal recessive disease where people who are homozygous recessive have this trait for the disease. Another example is PKU that we talked about, where if you have the recessive one, or recessive homozygous, what happens is the person itself has the disease itself. Another striking example is sickle cell disease for autosomal recessive traits, where the heterozygous parents are carriers and can pass on the mutated gene, but then the homozygous recessive ones have the trait itself. Like these two carriers end up with an affected male, and then it depends on like further generations if they get the trait or not. So if it's homozygous for them, they will actually do have the trait itself or be a carrier to it. And the effects itself kind of shows up in different generations, like it can skip a generation or two depending on how it was transmitted. So now carriers of sickle cell trait basically do not show symptoms of the disease, but they can also be protected from certain infections like malaria, where in areas where it's like a high prevalence of healthy heterozygous sickle cell carriers in certain parts of the world where infections like malaria exists. So basically it kind of helps with genetics costs, but like also genetic advantage as well, depending on the situation you are in being a heterozygote for sickle cell trait. Now we kind of go into the X-linked recessive traits. These are more prominent in males than they are in females. The one thing about this is that the trait is never passed from father to son because fathers give their sons the Y chromosome, not the X chromosome. Affected sons are usually born to carrier mothers because they actually get the X chromosome from the mothers. And about half of the sons of a carrier mother will be affected. And the trait often skips generations like a recessive trait would. And some examples include like color blindness and hemophilia. Now, there are some genetic notations when we have to consider X-linked recessive traits. And the first one is XX, where it's two X females with two chromosomes that are unaffected basically do not have the uh do not express the recessive trait itself. The XCX, basically the C here stands for carrier, so one is a carrier, one is not, but does not express the trait because it's basically um since it's recessive, it wouldn't show up in that female itself. Now, if there's two carrier Cs where it has the affected one, then the recessive trait is expressed and it's shown that it is, like in cases of like color blindness, then it would be there. Now, if there is an XCY, which is basically an X and Y is a male. And what we see is that with XCY, which is like the affected chromosome, where you end up having the trait in the guy because there is no X chromosome that can like take over. So one thing about the X chromosome is that one of them can be inactivated, and the other X chromosome can take over in females. However, in males, this is not a possibility. Now, XY is just a male with no affected X chromosome. It's good on all the color vision and all that with vision itself. Now there are X-linked dominant traits, which basically rarely skips any generations. And basically the affected male itself has all affected daughters, but no affected sons. Why? Because the affected male would only give the Y chromosome to the sons, not the X. And when it's dominant, it usually affects the X chromosomes within the daughters themselves. So that would be dominant in itself. And an example would be congenital generalized hypertrichosis. And that just basically just has hair all over, terminal hair all over the body, like this person here, where they have hair all over the place. So now to just like compare between X-linked recessive and X-linked dominant, it's more that with X-linked dominant, they are both males and females are equally likely to be affected, whereas with X-linked recessive, males are more affected. With X-linked recessive, the trait is never passed from father to son, but in X-linked dominant, the daughters are affected, but no sons. And then the affected sons are usually born from carrier mothers. The affected sons always have a affected mother. In X-linked dominant, about half the sons of a carrier mother will be affected. In X-linked recessive, it's also that all of the daughters of affected males and unaffected carrier females are carriers, and the trait skips generations in this recessive traits with X-linked. And then with about half the offspring are affected of an affected mother will be affected. Affected daughters can have affected mother or father depending on the dominance of that trait. And then this trait does not skip any generations. And with that, we will conclude our topic on inheritance patterns.